Okay, so in this section we're going to be looking at protein synthesis, and protein synthesis has two basic steps. The first is transcription, and the last, or second, is translation. Today we're going to focus on transcription and some differences in DNA and RNA that are important to this process. So first thing is, how do we access the genetic information in order to actually build a protein? Some things that you should remember is that a protein is made from a large number of genes, and that a gene is a section of DNA that has a specific code for a trait or a protein. When you look at genes can code for proteins, and they can code for some form of regulation of the protein. So not always the protein themselves, but you will always need the entire code in all of your cells. But part of that cell specialization was that idea that some genes were turned on, some genes were turned off in specific cells. This picture right here is very important because this picture, the entire picture, describes what's known as the central dogma. The central dogma. And so the central dogma is basically talking about the transfer of information. When you look at this image, what you see is initially you start with DNA. And when we want to make something like a protein or a trait, we need to start with the DNA, and from the DNA we're able to go through the process of transcription to form, technically, mRNA. Using information from the mRNA single nucleic acid sequence, we'll be able to go through translation and produce a protein at the end. When you're looking at a whole molecule of DNA, you should be able to recognize that different sections of the DNA represent different genes. And so the initial code is in the nucleotides. So the code for proteins are in the nucleotides of the DNA. And notice here that they also included replication, taking information from DNA to make more DNA. And eventually we can use that to make proteins or traits at the end. We had two scientists named Beadle and Tatum that discovered the idea that one gene codes for one enzyme, or one gene codes for one trait. And really, the main thing that we get from them is the idea that there is some organization of the genome. And the genome is all of the DNA of one organism. So we have the DNA of humans already mapped out, and we know that certain genes code for certain enzymes or proteins. And they'll be similar from one human to another. When you're looking at this image right here, this actually has to do with their experiment. Here they had different, basically what you're seeing is different groups. Your control group, which is your wild type. This is what was natural or nature. And then you have 3 mutants, which are your experimental groups. The main thing here is that this is a pathway of some enzymes and of some proteins that are being made. And it's basically saying that different or many enzymes are needed for the pathway to work. We can even say to be complete. So at the end, in all of these, we want to make arginine. And in order for us to get to arginine, we start with some sort of precursor. We need the first gene to produce an enzyme that allows us to make the next. And then we need a second gene, gene B, in order to alter this molecule into the next molecule. And then we need a gene C to finally alter that third molecule into our final product. So what they learned was that different sections code for different proteins, and that we didn't use all of the DNA for every single thing in the organism. That we had this idea of distinct units produce specific proteins, and that proteins can interact to produce one end product, which could be your trait or protein. Okay? Different sections code for different proteins. Distinct units in that DNA will code for specific proteins. Proteins interact to produce one end product, or can interact to produce a specific end product. But what we will need to look at next is RNA and the role of RNA in transcription and translation. What you want to keep in mind, though, is that in a protein synthesis, that protein synthesis is our goal, and that DNA has the code. So at the end, we want to make a protein, but initially we have to work with DNA. So RNA plays an important role in using the DNA code to create proteins. There's two major steps that we need to know. The first step is going to be called transcription. So this is our little overview of the process. In transcription, we have the DNA for one gene is going to be base paired to synthesize a copy known as mRNA. It's not really a copy because it's not exact, but it's a different form of that same message. The process of transcription is actually going to happen within our nucleus. So transcription is here in the nucleus. And so later on with transcription, this is the mRNA that we're making. That mRNA will move out of the cell, out of the nucleus, and move towards the ribosome for the second step, which is where translation occurs. Okay, so transcription in the nucleus, translation at the ribosome. So what happens in translation? Very simply, in translation, we're going to take mRNA code and we're going to read it to build the amino acid sequence. It does get read 3 bases at a time, and 3 bases on mRNA is known as a codon. So we're going to take the language of the nucleotides from the DNA, and we're going to convert it into the language of mRNA, and eventually produce the language called amino acids to make our protein. Another thing that we need to know is that there are 3 types of RNA. And the reason why it's called RNA is because of the sugar known as ribose. And the 3 types of RNA are known as, well, first of all, we have mRNA, tRNA, and we have rRNA. mRNA is going to be long and single-stranded, like you see in this image right here, the mRNA. tRNA, and I'm sorry, we want to add to that the mRNA has 3 bases called a codon. tRNA is going to be attracted to the mRNA, and its main thing is that it transports amino acids. And then you have rRNA, which is known as ribosomal RNA. This is transfer RNA. And this is messenger.
Mildred Guerrero, meet your ride outside immediately. Mildred Guerrero, your ride is waiting for you outside.
The rRNA, or the ribosome, will fold the amino acid chain. Or I'm sorry, it is It is folded RNA to make the ribosome. And it is the location of protein synthesis. So this image right here, if you can kind of follow what's happening, is really important. Basically tells us all of transcription, or at least the general idea of transcription. In transcription, we start with DNA. DNA is held within the nucleus of the cell. In order for transcription to occur, we need the DNA to actually open up, and we base pair A's and U's technically, and C's and G's, in order to make our mRNA sequence. Once our mRNA sequence is made and complete, we can actually leave through a pore in the nucleus. When we leave through the pore of the nucleus, our mRNA will actually meet up with a ribosome, which is a group of DNA and protein, RNA and protein. It's an open structure, not a closed membrane-bound organelle. But at the ribosome, 3 bases on the mRNA, which is a codon, will pair with what's on a tRNA known as an anticodon. And here, this little long pillar shape is actually a tRNA molecule. Notice that on the tRNA, we are also carrying an amino acid. An amino acid chain is being built, and this will eventually become our protein. It does have to fold first before that's possible. This would also be a good time to go back to your notes on proteins and the levels or stages of protein folding. But the last thing I want to talk about is some important differences between DNA and mRNA. Both of them, however, are polymers of nucleotides. Some obvious differences that you can see here is that for RNA, you have a ribose for the sugar. You have no thymine. You have uracil instead. And those are the nitrogen bases that we're talking about. And it's usually single-stranded, especially in the form of mRNA. When it's tRNA, it's going to look a little different. When it's rRNA or ribosomal RNA, it's going to be different again. On the other hand, for DNA, you have deoxyribose for our sugar. And this is a double-stranded molecule. And we have thymine for our nitrogen base. We're actually going to go ahead and stop there for today and talk about the steps of transcription in class.